In this video, we will discuss about the mechanism by which this TAGLN2 expressions on the cell surface of CD8 plus T cells goes down. So from the literatures, the researchers got an idea that it's the ER stress is somewhat negatively or irreverse is negatively related to this TALNG2 or inversely related to this TAGLN2 expression on the cell surface. So from that hypothesis they move on to check whether this is true or not, and if this is true, then what in the ER stress that causes the suppressions of this TALNG2 expressions on the cell surface. So first they check with this XBP1, which is one of the factors of the ER stress. Whenever the cell experiences ER stress, three predominantly reactions occurs. First one is this triggering of this XBP1, second one is this IRE1 alpha, and third one is this ATF4. So they check it with this XBP1 and they see that with the increase in the concentration of the ER stress, there is a increase in this XBP1 expressions, and simultaneously they see that there is a decrease in the TAGLN2 expressions. And they, in order to generate the artificial ER stress, they use three different reagents, that is DG, TG, and TM. Thepsigargin is TG and tunincamycin is TM. Not able to recall full form of 2DG, but all these three reagents cause ER stress, and with the increase in the ER stress, they see the decrease in this TAGLN2 expressions. And after validating that this TALNG2 expressions goes down with increase in the ER stress, they also check this observations using flow cytometers, where they see that with increase in the concentrations of this reagent that cause ER stress, they see the decrease in this TAGLN2 expressions. And these things are shown in the panel B, the flow cytometer results and the quantitative results for all three reagents, that is 2DG, TG, and TM, and the results are similar. That is, with increase in the concentration of this reagent, they see the decrease in the TAGLN2 expressions. And next, they also validated this thing using the confocal microscopy images, where we can see that when there is no reagents are introduced, we can see the presence of this TAGLN2 expressions. But with the increase in this tunincamycin, with the injection of the tunincamycin, that is 1 microgram per ml, we see the decrease in this TAGLN2 expressions. So after confirming that ER stress has a negative relationship with this TALNG2 expressions, they checked individually that which factors they checked individually, that is by knocking out this XBP1 or IRE1 alpha or ATF4 in the mice models and check the relative expressions of TAGLN2, and they find out that in the absence of IRE1 alpha, they see the increase in the TAGLN2 expressions, that is shown in the panel D. For the other two cases, they see the decrease in the TALNG2 expressions, and those results are shown in the supplemental figures. So in this figure, they have shown the results for this IRE1 alpha knockdown, and they see that when the IRE1 alpha knockdown, in the IRE1 alpha knockdown case, they see the increased expression of the TAGLN2 expressions whenever this reagents is introduced that cause ER stress. And these things they have validated using the flow cytometer also, where we can see that in the case of this knockdown, the TAGLN2 expressions are higher than the wild type. And from the literatures, they know that IRE1 alpha, once the IRE1 alpha activated, it will triggers the functional form of this XBP1. So they also check this thing using the knockdown of XBP1 and the wild type of XBP1, and they see that similar results, that is IRE1 alpha is high when the XBP1 wild type is XBP1 knockdown is present. That can be seen in this figure for different reagents. When the XBP1 knockdown is there in the mice models or in the cells, they see the high expressions of this TAGLN2. This indicates that it's the XBP1 that has a direct relationship with that has a direct relationship with this TAGLN2 expressions, as IRE1 first activates this XBP1 and then it activates the then it causes this down regulations of this TAGLN2 expressions. And these things, the results of this panel F is quantified in the panel G also, where we can see that with increase in the we can see that in the case of XBP1 knockdown conditions, with the increase in the dosage of this TM, they see that increase in this TAGLN2 expressions. And thereafter they validated these results by deactivating this XBP1 using two reagents, that is MKC8866 and KIR8, and they can see that in both the conditions they see the increased expressions of this TAGLN2 expressions for and simultaneously they see that reduced expressions of this XBP1s, because it has been inhibited by this their inhibitors. And in the case of stress reagent, they do not see a rise in the XBP1, whereas they see the rise in the TAGLN2 expressions. So this further validates their point that XBP1 has a direct relationship with this TAGLN2 expression, has a inverse relationship with this TAGLN2 expressions. And next they check the binding sites of this stress reagents with this transcription factors of this TAGLN2, and they are able to find the sites that coincides. They are able to find these binding sites between stress reagent release and the transcription factors on the TAGLN2 expressions, and that are shown in the panel I, where we can see that NOS XBP, that is CAG GTC, is in line with this human XBPs CAG GTC. And so after finding out some of the binding sites of the stress that can bind to the transcription sites of the TAGLN2 expressions, they wanted to check whether those binding sites get affected or not. So in the case where no stress is there, they do not see and they did this study using the chip QCPR. So in the binding site 1 and 2, when there is no stress, they do not see any increase in the relative occupancy, but when there is a stress, they see that increase in the relative occupancy significantly. And after validating that, they wanted to do further studies. So what they have found out is that NF kappa beta activations also has a overlap with the binding sites of the TAGLN2 expressions. So in this experiment in the panel K, what they did is they use this TALNG2 expressions with luciferase, and when they introduce this NF kappa beta, they see the rise in the luciferase expressions, indicating that TAGLN2 expressions are high. However, when they also inject this XBP1, they see that reduced expressions of this TAGLN2, indicating that XBP1 has a negative relationship, whereas NF kappa beta has a positive relationship with TAGLN2 expressions. And they have validated this positive and negative relations in the various kind of T cells. That is, in the case of the CD62 low and CD44 high cells, they see the high expressions of this TAGLN2 expression, high expression of this TAGLN2 when XBP1 has been knocked out using CD4 CRE. And similar results they have observed for the CD62 high and CD44 high cells. And these things are quantified in the right hand side of the panel L, where we can see that when the XBP1 is knocked down, they see the high expression of this TAGLN2, whereas XBP1 is present, they see the low expression of this TAGLN2. And these things in the panel L, they are from the panel L, they are observing these results. They are observing these results from the data that they have acquired, that the data that is publicly available about the ovarian cancer cells. So they are checking this CD62 low and CD44 high in the ovarian cancer cells or the tumor microenvironment of the ovarian cancer cells, where this representative cells are found. So they are analyzing the data that is publicly available in the about the ovarian cancer cells. It is in the tumor microenvironment or in the ascites of the ovarian cancer cells. So this is about panel L, and for panel M also they check it for other different conditions, that is CD62 low and CD44 high, and CD62 low and CD44 high. So, and what they have find out, they find out the similar results, that is in the case of XBP1 is been knocked down, they see the high expression of this TAGLN2, whereas when XBP1 is present, they see the low expressions of this uh, TAGLN2. So, and one more thing that they have find out is that in the ascites there is a low productions of low expressions of this XBP1 has been observed, and which is corresponds to high expressions of this TAGLN2. And in the panel L, in the panel N, they have presented a UMAP of the different kind of CD4 plus cells are found in the ovarian cancer patients. And when they check their ER stress relationship, they can see that ER stress signature genes are low in the area where in the area of the ascites of the ovarian cancer cells. And this is directly related to the other results, that is high expression of this TAGLN2 in those conditions which are shown in the panel L and M. And finally they check the relationship between XBP1 expressions and the TAGLN2 expressions, and they are able to find out that the XBP1 expressions has a negative relationship with this TAGLN2 expressions. And these things they have validated using the CD8+T-cells and also in the other cells, that is CD45+, CD19-, CD3+, CD8+ gated cells. So all this indicates that it's the XBP1 which is triggered by ER stress that cause low expressions of this TAGLN2, and that will cause the deactivations of these CD8+ cells. So I hope you understood that in the previous video they see why in the previous results we observed that CD2 plus helps in the lipid uptake. TAGLN2 assists FABP5 in lipid uptake in the activated CD8+T cells. So if this TAGLN2 get disrupted, then lipid uptake will not be happen and CD8+ cells will not be able to perform its functions for a longer duration of time. So in this experimental results they have find out a relationship or mechanism that why TAGLN2 expressions goes down. It's because the ER stress got triggered in the CD8+T cells whenever they come in contact with the ovarian cancer cells or in the periphery of the ovarian cancer cells. However, in the ascites of the cancer cells where XBP1 expressions are low, they see the more expressions of this TAGLN2, and their CD8+T cells are also found in the there are more abundant activated CD8+T cells found in that area where XBP1 expressions are low, and they can perform its functions in a more efficient manner. So that's all for this video. If you need to understand more about this experiments in detail and want to look on the supplemental figures, you can go to the reference that has been provided in the description. So thank you for your time.